Hi guys, this is the Medicine PG, and today I'll be covering my first video in a series of videos that I'll be making on acid-base balance, acid-base disorders, and ABG interpretation. So let us begin. So firstly, why is pH most important in the body? See, the pH, just like how our temperature is regulated, just like how our glucose levels or blood glucose levels are regulated, just like how our sleeping time, circadian rhythm is regulated, the pH is regulated along a very narrow range of 7.35 to 7.45. So why should our body have this regulatory mechanism? Why can't the pH be anything? So why? Because pH is responsible for a variety of metabolic and cellular functions. It maintains the normal cellular metabolism, maintains normal protein and membrane function, and any alterations in this pH from the normal values can cause our body to go haywire and lead to diseases. These diseases can affect our central nervous system, our cardiovascular system, and can definitely change how the electrolyte balance in the body. And so, what is the normal pH in the body? As I mentioned, the normal pH is strictly regulated around a narrow range of 7.35 to 7.45. So what can you infer from this value? It is slightly alkaline. Okay, it's slightly alkaline, the pH in our bodies. So how do we calculate this pH? The pH is actually mathematically it's negative log of H plus ion concentration in the body. So at a pH of 7.4, the H plus ions is 40 nano equivalents per liter. This is important to know. So the pH is maintained by a variety of different regulatory mechanisms, and there are three main mechanisms which regulate our pH. These are three lines of defense in acid-base regulation. First line of defense is the chemical buffer system. Second is the respiratory regulation, and the third is renal regulation. So let's begin with the chemical buffers. So the chemical buffers include both intracellular and extracellular buffers. The extracellular buffer is predominantly constituted by the bicarbonate buffer system, and the intracellular buffers are constituted by protein and phosphate. So I'll first explain about the chemical buffers before going on to respiratory and renal regulation. First, let's start with the bicarbonate buffer system. Firstly, I want you to know what is a buffer. A buffer is any solution that resists a change in the pH. So that is any change in H plus concentration, the buffer resists this change. Okay, so a buffer, the bicarbonate buffer system. So what is a buffer? A buffer is basically any solution that resists change in the pH. So how does it work? A buffer consists of two components. Consists of a weak acid, which is the bicarbonate ones. Sorry, which is the carbonic acid plus its conjugate base, which is the bicarbonate. So weak acid, which is the carbonic acid, and the conjugate base, which is the bicarbonate. That is the bicarbonate buffer system, what it consists of. So next, so if you see this equation, you'll be able to understand how it works. So basically this, I'd like you to split this equation into two. So this is one equation and this is one equation. Okay, that is, this is one equation, this is another equation. So the H2CO3 is the common part between both sides of the equation. So first, let's see this part. So this part usually happens in the lungs, and this part usually happens in the kidneys or the metabolic aspect of the body. So we'll start from backwards. So H plus combines. So say there's in the body, there's an increase in acid production. That is, there's an increase in H plus production. How does the body respond? The bicarbonate buffer system, bicarbonate combines with H plus to form carbonic acid. So now H plus is increased. Now carbonic acid is increased. So what happens next? So this carbonic acid eventually causes an increase in pH, right? So our body has to maintain the pH to a normal level. So it breaks down this carbonic acid. So the reaction then goes this way to form CO2 and H2O. So CO2 goes to the lungs and it is exhaled out of the body. So this way, an increase in H plus does not cause a rise in pH, as eventually the carbonic acid levels remain the same and the H plus levels are buffered. So vice versa, say H plus levels decrease or bicarbonate levels decrease, then the reaction will go the other way. So CO2 that is formed will combine with H2O, form H2CO3, which will dissociate into bicarbonate and H plus. So this is how the bicarbonate buffer system works. Depending on whether the acid production increases or decreases, there will be a shift in which the direction the enzymatic reaction goes. So one more thing that I want to mention here is the concentration values of the system. So you have carbonic acid, H2CO3. The normal value in the body is 1.2 milliequivalents per liter, and bicarbonate, HCO3, is 24 equivalents per liter. So why is the value of bicarbonate almost 20 times, or actually 20 times that of carbonic acid? This is because the body has something called an alkali reserve. This alkali reserve is the excess of bicarbonate such that it can, in case of any scenario where there's a sudden increase of H plus or acid, this excess bicarbonate can be used to immediately buffer this H plus and make sure the pH does not change drastically in a short period. So that is called the alkali reserve. This system, it acts very rapidly. It's a short-term system, and the phosphate and protein buffer systems, as I mentioned previously, these are intracellular systems. So the phosphate system is mainly constituted by sodium hydrogen dihydrogen phosphate, and the protein buffer system is made up of hemoglobin and plasma proteins, especially albumin. And one point of interest is in the hemoglobin, the histidine's imidazole group is responsible for the buffering action. So next, respiratory regulation. This is the second line of defense. It acts within minutes, and even this, it's not a long-term mechanism of maintaining the pH. It is relatively short-term. So how does this work? So as you can see in the diagram, whenever the pH increases or decreases, there is a change in the respiratory rate. Okay, so that's, as the name says, the respiratory regulation. There's a change in the respiratory rate depending on the change in pH. So firstly, if the pH decreases, that is called acidosis. It stimulates the brain and arterial receptors. These are the chemoreceptors. So in acidosis, there's an increased respiratory rate. Whenever you breathe faster and harder, there's a CO2 washout. So when the CO2 washout occurs, the value of blood CO2 decreases. As you remember in that carbonic anhydrase reaction, when the blood CO2 decreases, then the H2CO3 will also decrease because the reaction will go the opposite way to generate more CO2. So carbonic acid value will also decrease. When the acid value also decreases, so the pH becomes more alkaline. So pH increases. So you have just compensated a decrease in pH by an increase in pH and restored the homeostasis. So that is how the respiratory regulation responds to a decrease in pH or acidosis. So vice versa, in case of alkalosis, the respiratory rate will decrease. So what happens in alkalosis? pH increases. So when the pH increases and the respiratory rate decreases, so that will lead to accumulation of CO2 or CO2 retention. This CO2, since it's high, will combine with H2O, and carbonic anhydrase will facilitate this to form carbonic acid. Since the value of carbonic acid increases, the pH will then decrease as it becomes more acidic. So now you have neutralized the alkalotic change in pH by decrease in pH and made it acidic. So it's become pH has increased and you've decreased the pH back to normal. So you've restored the homeostatic mechanism. So this is how the respiratory regulation works. Next, we'll go to the renal. So next we have the renal regulation. So this renal regulation is the ultimate final line of defense for all regulatory mechanisms. It is the final one. The only disadvantage about this is it takes hours to act. It takes quite a long time to act. So initially it's dependent on chemical buffers and the respiratory regulation to maintain the pH. But once it acts, it is a long term, acts for long term. So how does this work? So it works in three ways. At three different parts of the tubules, it works differently. So first in the proximal convoluted tubule. So at the PCT, it regulates the acid-base balance by doing two things. One, excretion of H plus ions. Two, conservation of bicarbonate ions. So we'll split this. First, let's start with excretion of H plus ions. So H plus in the cell, which is formed from carbonic acid, the same reaction. So CO2, I'll just describe this again. So CO2, which is formed during the normal cellular metabolism, combines with water to form carbonic acid in the presence of carbonic anhydrase. This is carbonic anhydrase type 2 in the cell, in the proximal convoluted tubule cell. So carbonic acid then dissociates to form H plus and bicarbonate. This H plus is secreted into the lumen of the PCT by sodium hydrogen antiporter. So sodium enters and H plus goes out. This H plus actually in the diagram it's showing that it combines with bicarbonate, but actually H plus that is secreted from the cell is excreted out in the urine. So that is how the excretion of H plus occurs. Next, we'll go to how the conservation of bicarbonate occurs. So again, starting from this step, now bicarbonate CO, so HCO3 is filtered from the glomerulus into the lumen of the proximal convoluted tubule. It is present in the filtrate. So this bicarbonate in the filtrate combines with H plus, which is also secreted in the lumen. So this combines with this H plus to form H2CO3, and this H2CO3 dissociates by carbonic anhydrase enzyme. This is type 4 carbonic anhydrase to form H2O and CO2. This CO2 goes back into the cell, and as you can see, this reaction occurs again. So this CO2, HCO3 from here finally goes here, enters the cell, and it finally is transported from the cell into the bloodstream with the presence of Na plus. So the next compound is sodium bicarbonate. It goes into the bloodstream and H plus is excreted into the lumen. So this is how renal regulation at the PCT works. So next, at the distal convoluted tubules. This occurs at in the alpha intercalated cells of the DCT. So what happens here is sodium disodium hydrogen phosphate, which, as I mentioned earlier, it is part of the phosphate buffer system. This is secreted into the lumen of the tubules, and it breaks down to form 2Na plus and a HPO4 2 minus. So this Na plus, we can split it as one Na plus, one more Na plus. So what happens here is, so again in the cell, the same reaction occurs, forming bicarbonate and H plus. So this H plus is transported out with the sodium hydrogen antiporter uh, into the lumen, and Na plus goes back into the cell. So this H plus is now present in the lumen. This H plus combines with the HPO4 2 minus to form H2PO4. H2PO4 is known as phosphoric acid. This phosphoric acid then combines with the extra Na plus from here to form a compound known as sodium dihydrogen phosphate. So this compound is called as a titrable acid. And so by excreting this titrable acid, the urine becomes acidified. So the normal blood pH is 7.4, but the normal urine pH is around 6. So how is the urine so much more acidic than the blood? It is by this mechanism. The DCT cells, they secrete a titrable acid known as sodium dihydrogen phosphate into the lumen, which is excreted into the urine, and that is how the body clears the acid. That is how the kidneys clear the acid from the body. So third mechanism is by excretion of ammonia. So this mechanism, the first compound that gives the ammonia is glutamine. So glutamine, by glutamine A's enzyme, breaks down glutamine to glutamate and ammonia. So glutamate again is broken down by oxidative deamination to form alpha ketoglutarate and ammonia again. This alpha ketoglutarate goes via the TCA cycle to form CO2 and H2O. Again, this whole carbonic anhydrase reaction repeats. Finally, it forms bicarbonate and H plus. So H plus again is secreted from the cell into the tubular lumen. H plus, and along with H plus, ammonia is now secreted from the cell into the filtrate. So H plus combines with ammonia to form NH4 plus. NH4 plus is now positive. It cannot be secreted. It needs to be combined with one more negative compound. So this A minus is the Cl. Chloride ions combine with NH4 plus to form NH4Cl ammonium chloride, and this way the body excretes ammonia in the form of ammonium chloride salt. So this is all for the first part of acid-base balance. So I've covered the biochemistry and the physiological aspect, and the next part I'll be discussing more about acid-base disorders. Thank you very much.